the next lecture is just an introduction about what are the parts in the microscope that we will be using or learning in the next couple of days, and why I think microscopy is important. Biomedical research is obvious, and all of you are already probably users of some way or another of fluorescent microscopy. But basically, to summarize, nowadays we can go from microscopy from samples that can go on the millimeter, centimeter to down to the nanometer, and even more. In the last couple of months, there were papers where you can localize a molecule with an Armstrong resolution if you have enough photons and if you have something white that can give you from the same point, enough photons. So microscopy has become the really fundamental research infrastructure and technology for the discovery of new biology. But what is a fluorescent microscope, and what are the difference between epifluorescent microscopes and confocal microscope that we will be using for most of our methods? Well, in a fluorescent microscope, epifluorescent microscope, wide field, we have a lamp, which is basically the source of light. And then we have a block, which is used or shown here, that split excitation and emission light. And this dichroic mirror, we will see at the end of the talk, has some characteristics that enable us to do this. And then we have some optics in order to focus light on the specimen. After the specimen, as we learned from Dave, absorb the light, it emits at longer wavelength, and basically some of the light is also picked, some of the fluorescent light is also picked by the lenses here, and is conducted through the dichroic mirror, pass through the dichroic mirror, and then we can have either the camera or our eyes to check what is the fluorescent. And this is basically a very simple optical diagram of the epifluorescent microscope, and basically you see here the source of light, and then the dichroic mirror, the objective or the lens that focus the light. And one thing that you can figure it out here is that if you have light focusing on different depths, this will give you information in your detector with different depth too. And this is the reason for why when we watch a epifluorescent microscope, we have some blur image, right? When we speak about confocal microscopes, what we have is more or less the same. We have detector, we have light, we have optics, we have dichroic mirrors, but we have one optical objective here, which is important, and it's called the pinhole. And the main purpose of this pinhole is that we basically reject light which is not in the focal plane that we want. So we focus the light before the detection, and we make the light pass through this little hole, and all the light that is not right in the focus of this hole will be rejected. And this is the reason for why we cross-section a slide of our sample with the confocal microscope. There are other parts which are important for confocal microscope, like the scanners or the stages, because contrary what is happening here, that we can see all the pixels in the same plane, in the confocal microscope, we need to go one by one, and we need to recover the image. And it's more or less, if you have, this kind of eyes, glass lenses that are used for anti-fatigue or prevent myopia, where you have many pinholes. If you look through them, you will see an image that is composed of little pixels, right? And I like the correlation with that. But well, with the wide field, eventually we can have images like that. When we jump to the confocal, you see you increase a lot, the signal to noise, and you decrease the blurring. And there are many different ways of doing confocal microscope. We will learn that laser confocal microscope is one of the simple, but then you can have spinning disk with many pinholes, or you can have multiphoton microscopy. And multiphoton microscopy is a way of doing confocal microscopy. And then you can have so many other techniques that we will discuss later. So what is important, or what are the key parts for a microscope? Well, we need light for sure. We need something that moves the light. We need detectors, and we need some other parts, optics, electronics, that are important to improve or to split or handle with the light. So we will try to cover all of this along the lecture. So speaking about light, when we speak about confocal microscopy, what we have is, lasers. And most of the lasers using confocal microscope are continuous wave, which means that if you measure the intensity over time, it's more or less constant. So you have more or less average amount of light, and you have some other, optical tools in order to attenuate light for, the imaging. But the output of our lasers are always more or less at the same range. And this can be, gas or more modern solid state lasers that can have, many, many bands. Most of these are discrete, so we have single wavelength. And, in all these cases, as I mentioned, this is basically a continuous wave. When we want to do film, we need pulses. And we will see tomorrow what it means. Film is fluorescent lifetime imaging. And for fluorescent lifetime imaging, we need to excite our sample and then wait for the photons coming from the excitation. And this is when we start to use pulse lasers. And pulse lasers, there are many. This is one of the examples that we have now in the market, the white lasers, which are very interesting because while they are fundamental, modulated at usually some frequency from, I don't know, megahertz frequency, and they can cover very long range of excitation from 390, 400 to above a micra, so 1,200, 2,000 nanometers. And basically, instead of having a continuous amount of light, you see that we have a short pulse and then nothing, and short pulse and then nothing, short pulse and then nothing. And we take advantage of these windows because if you have detectors that can detect the photon coming after excitation, well, we can do lifetime. But what else is out there? Well, we will see and we will, do practicalization with the pulse laser and multiphoton microscope. And basically, these lasers are very powerful. And basically, there are many in the markets. These are, 4 brands that I want to highlight. Particularly, I want to mention that this is the laser that we will use in our practicalization. You see here, and this other is also in the unit, but in the hospital with another microscope. And all these lasers can pulse at 80 megahertz, 50 megahertz, depending on the configuration. And these 3 can tune between more or less 680, 690 to 1080. So you can excite, fluorescent molecules that usually are excited in the visible or in the UV by 2 photons of the half of the energy. And I will explain in a minute what this means. But one of the powerful things of this is that confocal, 2-photon microscopy is intrinsically confocal. And what is this? Sorry, before to go that, there are also new lasers that are, has broader band. These are extended, to the 1,300 nanometer, like the Insight or the Coherent lasers. And they are very powerful because you can go even farther and you can use, farther, fluorescent molecule, red fluorescent molecule, which needs more than 1,000 nanometers to be excited. But what we mean for, 2-photon excitation and what is the principles? Well, we deserve 2-photon excitation to basically Maria Gopel-Meyer, who in her PhD thesis in the 30s, she described this formula that give all the fundamentals of 2-photon excitation. And the formula tells us that basically, if you want to have 2-photon excitation, you depend on the power of 2 of the power you are applying and the frequency and the lifetime of the molecule. And if you have enough power, and if you have, a good lifetime, and if you have pulses that are, short enough, you will be able to do two-photon excitation. And if we think about, Parrinchia velocity diagram, when we excite, a molecule, right, a fluorescent molecule with, 400 nanometers, we will move our electron from the ground state to excited state. After some, excited state conversion, it will fluoresce. And the key to understand how this is possible, that two photons of 800 nanometers, so half of the energy, can excite and the same electron and do the same circle. The key of this is, in part, the excitation time. So excitation time is a process that happens more or less on the femtosecond time scale, while fluorescence is in the nanosecond time scale. So what we need is basically, instead of have, light, in a continuous amount, we need to pack our photons in a range which need to match this number, so femtosecond, and all these lasers are, femtosecond lasers. So all these lasers has pulses that are dispersed with behind of 100 femtoseconds. And if we can put this laser to all the photon pulses in this little burst, then we can focus them with optics on a point, and in here the magic happen, and we can have two-photon excitation. So finally, to have two-photon excitation, we need basically, putting together in time and putting together in space two photons of the half of the energy that we use in single photon excitation. And one amazing thing of this is whatever you have above or behind this point, it will not be excited because you don't have the two conditions, temporal compression and spatial compression, which is the requirement to have two-photon excitation. But what is so unique about two-photon excitation? Well, scattering depends in a way of the inverse of the power of 4 of the wavelength. So as far as we go, this is a logarithmic scale, and this is scattering. You see that it decay a lot, the scattering, while we go farther in the excitation wavelength. And there is a window here that we call two-photon window, in which basically most of the absorbers we have in the tissue, hemoglobin or melanin, has some sort of a depth. And I insist that this is an exponential, it's a logarithmic scale, so you decrease order of magnitude of absorption here. So when we have two-photon excitation, scattering and absorption are decreased at farther wavelength. So this is the reason for why two-photon excitation can penetrate more in the tissue, and we can do images at, 1 millimeter on depth now in natural condition. And another important thing is, as I mentioned, is the autofocus, and this little dot here is the two-photon excitation. This is basically a cuvette where we have, rhodamine 110 is a fluorophore, and if you do excitation with single photon, you see, the cone of excitation here. But with the two-photon 780, you see that you can only excite in this spot where you have time and spatial compression of the pulse. So basically, you don't have autofocus, and you reduce a lot photo bleaching because you don't have any excitation above or after this point. So this is also very useful for science, because you don't want to bleach fluorophore that you are not imaging, right? Any photon which is above or behind the focal point, we will not take care of them because we will have a pinhole to remove it, right? Okay, so when we talk about laser scanning microscope, that can be, just a single photon excitation as a confocal, or STED microscope for stimulated emission depletion, which is a super resolution or multiphoton. What we do is basically we use scanner that move the beam in the sample, and then we have optics to split excitation and emission and detectors. These are two of the commercial confocal microscopes we have in the unit, and we will use it for the practical session, the 800 and the 880 from SAIS. We have this year a demo from SAIS, as I mentioned before, which is the 980, and we will be using. It has also some peculiar detector, like the ID detector, that we will also take advantage. And then this is the 2-photon film microscope that we call Diver, because this stands for a particular detector that was developed by Enrico and Sasha here, and is particularly interesting for imaging on in vivo samples without any clearing, so in native condition with highly scattering, and is really, really sensitive in comparison with any other 2-photon microscope. So what we mean by scanner? Well, the scanner is the two mirrors that can move with a motor, and if you combine these two mirrors in a particular way, we will see in the practical session, you can rust. You can go through all your samples, and then you can recompose an image. What is interesting about that is fast and is stable. Your sample is not moving. You are moving the beam, right? There are other methods like galvanometry scanner, in which you have a resonance scanner that go as a high frequency, or you have galvanometry scanner are the one that I mentioned before. Resonance scanner are the second that I mentioned, and then you have a scanner that can be used with some optical element like prism, this polygonal scanner, or acousto-optic reflector, because if you have a crystal and you apply radio frequency to this, you can change you generate greetings here. You can change how the light pass through this optical object, and basically do this in certain way. You can even use this to scan. We use it most of the time to attenuate our lasers, but we will see in the Diver, because it's open, so we can go and look by part by what they are doing. But also you can use stages. So you can use, instead of moving your beam, you can move your sample, which is good. They are very precise. They are very expensive, usually, piezo stages. But basically, it's very useful, and we will see in some of the application that Fernando Stefani will tell, they use stage for movement, because they want to have very, very precise position for super resolution techniques that want to achieve, I don't know, sub 10 nanometer resolution, where you need to have really high precision. When we speak about confocal microscope, there are two kind of configuration for the detection side. In the detection side, you can be on the descan mode or non-descan mode, and I will explain in a minute, and you will understand easily what they mean. So we have light right here. It can be any laser, can be 2-photon, or can be visible lasers, and then we have an optical element here, dichrom mirror, which will reflect excitation, right? And then we will have our scanner, and then some optics. And this optics is important to basically scan our light across the field of view. So every microscope, every fluorescent microscope has the tube lens. And when we want to do scanning, we have to have a scanning optics, scanning lens, that enable us to conjugate these two focal planes in a point and have the distance to the scanners in order to move the beam here. And then this guy will make the shove to put the light back on the objective, doing basically the scanning without getting out of focus, right? This is the way that this works. When we use descan mode, the light goes through our sample, and then the same optics that focus the light will pick the fluorescent, and the lights come back. And in here, it will split between excitation and emission, and will reach our detector. When the light reach to the detector, in this configuration, the light is quiet, is collimated, is not moving. And this is the reason for why we can use this configuration for regular confocal microscope. We can focus again the light, pass through the pinhole, and avoid any focus that is in any point in the field of view that is out of focus. And this is only possible because the beam is quiet, right? It's not moving. This can be done, with confocal or two-photon excitation, but there are some other ways to make confocal microscope. And this is basically, by the way, this is the configuration that most of confocal microscope uses, right? Commercial confocal microscope. In non-scan mode, it's only a possibility when we are working with two-photon excitation. And you will see in a minute why. In the non-scan mode, we have our laser, and the light goes again through our scanning optic and the sample. But we can pick the light before going back to the scanner, which cancels the movement of the beam, and we can pick the light here and focus the light in our detectors and form an image just from the volume that two-photon excitation is given to here. And why? Well, this is possible because in two-photon excitation, we have intrinsic confocality. We don't need a pinhole. And this is very useful because in some situations we have such a weak, fluorescence that if we waste most of the light passing through all this optic, you compromise the capability of doing images. And we will see in our diver microscope is this configuration. We have, non-scan. And for those of you who will be interested, we can visit the hospital because in the hospital, the other microscope is built in D-scan mode. So we have 1 and 1, and you will see how each of the configuration look like. But this is a very useful, approach for many of the two-photon excitation microscope. Okay, speaking about, scanning scanners, the galvanometric scanners are, this kind of, optical elements, which has two motors that can move, independently these two mirrors. One scan in X, the X axis, and the other scan basically the Y, and then you do, the scanning and you form an image in your microscope. The advantage of this is a very high quality, is high resolution, and the drawback is, you need to go point by point, and of course you compromise time. So it's slow that the time that take a camera. In the time that more or less you take an image in a camera, you will have few pixels scanned by the scanning lens, but not a full image. Now there are other technologies like the spinning disc, in which you can have a wheel with many holes, and you can put this wheel on front, of some optics, and then you can project the light in the chip of a camera. And basically you collect many pinholes, and if you are moving this, you renew this all, a very high frequency, you coordinate this frequency of rotation with the frequency of reading of the camera, and you can make an image in a confocal resolution at very, very high speed, and with very good resolution in C2. And this is a very nice development that we have, if you are interested in doing, very quick or very fast, acquisitions for processes in cell or in animals. Okay, so we have the light, we have the scanner or the stage to move the beam, and then we have to detect the light. And about detectors, we have many different kind of detector, but for point, scanning microscope, PMTs and avalanche photodiode are probably the most used. The PNT, these are the one that we have in the epi detection of the diver, and this is the one that we use for the diver detector. They have many different, surfaces that can be sensitive to different ranges. And this is, for example, bialkali or multi-alkali. These are the most common detectors still in the market for confocal microscope. But then the most modern are the gallium arsenate phosphidine, and they are very sensitive. You can get up to 30, 40, 50% of quantum efficiency. And they are sensitive basically in a range that goes usually depending on the surface from, ultra UV to infrared. The avalanche photodiode has some advantages, and in particular, you have, big ranges. But the big advantage is the sensitivity. You have really an increased amount of sensitivity of how many photons you can detect, and we will see why. And the other important thing is that in comparison with the PMTs, signal here is digital and signal here is analog. So if we want to do some of the technique that we will be doing, like, FCS, or we will want to do FLIM, we need to do digital signals. But how PMTs work? Well, usually we have, a photocathode and then photoanode. And between this, we have usually power, voltages. And then we have, these diodes change that usually are 8, can be different, but usually are 8, in which basically we produce this amplification from one photon to many photoelectrons. So usually for one photon that hits, the photocathode, we amplify, like 4 to the 8, so 66,000 photoelectron for each of the photon that we get in the image. And the interesting thing is, well, we can always put more voltage here, but as much voltage we put, we will start some releases of photoelectron from the diode. That doesn't mean that are coming from the photons that hit the photodetector. So this is noise. So we will play around in microscopes. If we play with more gain or less gain, you will see that you will get more counts, but at the end you will get a histogram which is not packed. It's really noisy because you have many photons which does not belong to signal. But the good thing about this photodetector is that you have, very, decent sensitivity, and you can read them very, very quick in nanosecond time scale. So these are useful, very useful for fluorescent lifetime image. And the other important thing is they have very large areas. So focusing light here is quite easy of focusing light in APDs because they are very, very small, areas of detection. In the APDs, in the average photodiode, we don't have this cascade of amplification. This happens because we apply a voltage between, this doped region and the detection. And as much power we apply through here, you will see that we can go from, from gain of 1,000 to 2,500, and we can increase, amount of, signal quite heavily. And this is because every photon that hit here can produce many more photoelectrons, and then this is translated immediately to the cards or the electronics, and this is connected to the occurrence of single photons. So basically, it's a intrinsic, digital detector in comparison with the PMTs. Nowadays, there are combinations of these hybrid detectors in which there are combination of amplification and EPDs, and these are very nice. We use it for lifetime in the LFD with Enrico for many applications. They are very useful, extremely sensitive, and the bad thing is they are quite expensive, and they can cost probably 2 or 3 times of a PNT or 2 or 3 times of APD. So they are quite expensive still nowadays. But if we wanted to do imaging, not only in a point, but also in a chip of a camera, there are many options nowadays with cameras. And when we learn about light sheet microscopy and some of the super resolution technique along the course, they rely on cameras. And camera, there are many options like CCDs, ICCDs, EMSDs, or CMOS camera. We will see in a minute. They are all used in different applications. And there are a group that I want to highlight here, which are not cameras, they are PMTs, but they are arrays of PMTs that in a way we use it as a camera. And SAIS has released this probably 5 years ago, this array, which is the ID detector, in which they use, a detector like this, 32 channels, in a way that they spread this spatially, and they can, in a way, image the PSF, and using this, detection, they can improve resolution. And there are some others detector like the 5x5, 8x8, or the SPAD array that Giuseppe Bicci-Domini will tell you more tomorrow, that can very improve the capability of doing imaging in something that is now known as image scanning microscopy. We don't scan a point, we scan an image, and this image is used to improve resolution, to do some other things like correlation, etc. So it's a powerful technique that enables us to do many more things. When we compare, CCDs with CMOS, the most important part here is, well, they all have, independent actuators, but the main difference between these two configuration, the charge coupled device or the complementary metal oxide semiconductors, the CMOS or the CCDs, is how we read the data. In the CCDs, if a photon hit here or here or here or here, we need to wait until the information is passed through, the readout, and one single charge to voltage converter is, doing all the job for many pixels you have in an image. And this is good because you have the same noise in the CCD pixel by pixel, but it takes a lot of time to collect an image because you have like a very small, entrance to your, electronics, because you can do this only one by one. With the CCDs, the thing is that we have a column of amplifier and we read lines after lines, which make way more faster. And the drawback is now the noise pixel by pixel can change. And we have a more noisy, image that needs some other handling, with the data. But these are, a fair comparison between the two. And you see that, even CCMOS, if they are good, and now they are getting very, very good, you can get really decent images. And this is one of the one that we use in the LFD many times for many of the paper, as some of the results that I will show with the light sheet microscope, I think Wednesday. But if we want to go to single molecule detection, we need to count photons from a single emitter. And in that case, we need to have, readouts that can handle with very, very, very low noise and very, very few photons hitting on the detectors. And these are, the show of the EMCCDs. And most of the EMCCDs relies on a CCD architecture, so it's the same of a CCD. But after that, they have an amplifier. And in this amplifier, they can, manage to improve the signal to noise in order to have, better images. And this is the same comparison than before, but now we can see in comparison with the back illuminated EMCCD, and you see that the signal to noise improve way better than the others. And these are the technology that, most of the microscope for super resolution based on camera use EMCCDs. While I have to say that in the last 3, 4, 5 years, the CMOS, the scientific CMOS, are getting so much better, and they are so much cheap, and the chips are much bigger, many more pixels, that they are, co-opting the field of super resolution. Okay, what else we need for microscope? Well, we need electronics, and we need some other elements like filters and wheels and some mechanics in order to make it work. So for moving the scanner, we will see in the lab that we use, some boxes that have the drivers, and these are the guys that give the information to the scanner. So if we apply a voltage, they will tell to the scanner how much they have to move in one direction, another, etc. And if you can control of this, you can do raster scanner, you can do lines, you can do circles, and you can do any shape you want. And this is how, when you go to a party and you see a laser doing weird things, well, they are using scanners to do these things, right? So most of the focal microscope use this technology, and we always also need an interface. We make the conversion between these two things. For fluorescent lifetime imaging, we also need cards that can count photons. But before counting photons, if we have a detector which is analog, like PNTs, we need to condition this signal. And basically, we rely in amplifier and constant fraction discriminator, which is basically a device that can translate from an analog to a TTL signal, so to a digital signal that is used in the car to measure the lifetime. And we have Alessandro here who brought amplifiers and CFDs and film cars, so we can sit with him and learn in the detail how all these things works if you are interested in. Then we have optical filters, and in optical filter there are a huge amount of different options, but the most common use in microscopy are the interference filters, where they deposit layers of different material, and they basically have interference in a way that can allow the light to pass or not, and they are very sensitive to angles. So you can make interference filter for dichroic 45 degrees, but 0 angle or any other. And this is important because if you use a filter of interference filters for light which is not coherent, it will not work. And we will learn from Sasha that for the diver detector, glass filter that absorb light, they are just transmitting light because they absorb the light, and whatever is not absorbed is transmit, is the key element in order to do imaging in this microscope. And finally, I want to cover a couple of new things that are important because they are taking more and more interest in the community for deep tissue imaging, which is the adaptive mirror or adaptive optics. And this is a problem that we have even in the two-photon excitation. When we have light focused on with a lens, when the light gets into the sample, if the sample has changes in this refraction, different composition, etc., the photons that came from this place or from this other place will have different path. And at some point, it will not focus on the same focal point. Because the front, if we think of a pulse of light as a front, right, it will be basically distorted. Well, if we have something, some sort of compensator in order to pre-compensate how we want to bring the photon to the same point, well, we will improve this situation. So this is a 3D zeta stack that we did together with Sasha many years ago. This is lung. What is black here is air. So this is a very tough sample because you have tissue and then air. So the most high change in index of refraction. So as soon as you go deep, the signal is very high, but the resolution of the image is very poor because the light is getting not focused in the same place and the PSF is getting distorted. So the wavefront is being affected. So nowadays we have these, little mirrors, which are composed of many small mirrors that you can accommodate, and you make, the beam pre-compensated in a wavefront to basically get to the same point, regardless from where you are coming to the sample. And this is a thesis from Simon Limans, PhD student of Enrico, where he managed to image, neurons at very high resolution, up to 2 millimeters in the brain, without cleaning or without any, removing of autofluorescence or etc., which is very tough or very challenging to do. So to close, I want to introduce what are the custom technology we have in the lab in here. So this is a microscope, a 2-photon microscope built on top of an icon body, and here is the scanning cage. So we have scanning optics. So basically scanner, scanning lenses, and then we have detectors. And in our configuration, detectors are in desk-cam mode in this configuration. So the light passes through the scanner, and we have a fiber-based 780 2-photon laser, which enable us to do 2-photon excitation. And then the many other things were built by us. We got the parts and for the cars, etc. So it's a microscope that we can open and look inside and see what are the parts there. And the second interesting microscope is the diver. The diver side is still under construction, is this detector, but the epi detection is already working, and we can do film and we can do imaging. And it's a very nice microscope that we can go in and see all the parts. And in this case, you see that the detection is right up after the dichromatic mirror, which is here. And we have two detectors here in order to do, imaging in 2 channels. With that, I want to thank everybody, say sorry for all the inconvenience with the first presentation. And I want to thank Hontao because this is a lecture that I reuse many of the slides from him in the LFD. And again, I want to thank many of the agencies that support the developing of the workshop and also the research in the lab.